Okay, so this week we'll start our conversation about mutations. Today's part 1, next Tuesday part 2. And as usual, I'd like to start with a simple question for the whole class. What are mutations? If you don't want to yell it out, just talk to your neighbors, but what are mutations?
A change in DNA sequence.
Change in DNA sequence. Change in DNA sequence. Anyone else? And there's many right answers to this question.
Beneficial, detrimental, or neutral change.
Change. So beneficial, detrimental, or neutral. So any change, right? Any change. That's not much different from this. Change in DNA sequence. In a way, by saying change in DNA sequence, we're not making any judgment as to whether that change is for the good, for the bad, or for the no change, right? Or neutral.
Spontaneous error.
Spontaneous error. Spontaneous error. So I'm going to spontaneous error. What else? What other things come to mind when we say mutations?
It can affect the phenotypes.
Can affect the phenotypes.
So like appearance.
Yeah, so I like that. I'm just thinking the technical phrasing. So we'll you say affect phenotypes. I'm gonna say phenotypic effects. Oh, my writing looks huge here. Wait, that's better, I think. What else? There's still more discussion here. What are we thinking? I don't want to put you in the spot, but I'm going to put you in the spot. Anything else, anyone? Yeah.
Michael, what was just about variability?
Variability. So, variability. Any other things? All right. So, love this. What we're going to do is we're going to go down the list and discuss how all of these, in a way, are correct, right? And talk about some of these things in further detail. So, changing DNA sequence is ultimately the textbook definition of what a genetic mutation is, right? And that right off the bat clears the air on some things like a mutation necessarily has to have some sort of detrimental effect. I know that you very strongly emphasize that a mutation can be detrimental, beneficial, or neutral, right? But so both of those together already kind of clear the air a little bit with regards to that. There's this relatively common misconception that mutation equals bad. Mutation equals something negative, something broken, right? We'll circle back to that in a second, though. But technically speaking, a genetic mutation is simply a change in DNA sequence. Okay? Now, I'm going to jump to phenotypic effects. It is true that many mutations have phenotypic effects, and it is also true that it is because of those phenotypic effects that these mutations may flourish in a population and eventually overtake all other equivalent sequences in a species in the process of evolution. It could also be that that genetic mutation gets wiped out by natural selection quickly because of those phenotypic effects. But super strictly speaking, and again, circling back to this idea that certain mutations can be neutral, mutations do not have to have phenotypic effects. There are many, many, many genetic mutations that just don't manifest themselves physically in any way. Okay? So we kind of go back to this idea that a genetic mutation is simply a change in DNA sequence. Spontaneous error, I like that because it brings up two emphases on two different concepts. One, the spontaneity of mutations, right? It is sort of accepted that even though you can mutate, you as a human being can mutate a genetic sequence with technology, and now with these days with CRISPR and genetic engineering. The truth is that for the history of life on Earth and a good chunk of the history of molecular genetics, you can expose a living organism to a mutagen agent, and even then mutations will be spontaneous. You have no control over them, right? So spontaneous / random. But I also would like to emphasize error, because error can be understood a number of ways. One is it's just different from what I expected, right? So an error is simply something different from what we expected to see here. But it also has that sort of negative connotation. If I say, oh, you committed an error, or you made a mistake, right? There's that negative connotation that now things are bad. Not necessarily. Many mutations actually have beneficial effects, and that's how they became selected for throughout evolution. And last but not least, the variability. This idea that mutations are ultimately the source of variability, biological variability in life on Earth, right? This idea that it is genetic mutations that generate the kind of variability that allows for evolution, that allows for adaptations, and so forth. Right? So all of these are correct, but we are going to, as we go along today, we'll come back to some of these points. So in general, mutations are changes in DNA that, and here's where we start going a little into the weeds when it comes to definitions. There are changes in DNA that are rare enough in a population, and the most people will agree less than 1%, some will say less than 0.1%, or that cause a specific molecular dysfunction. Why am I emphasizing that? Because culturally, we cannot shake off that idea that if there is a genetic change, if there is a change in genetic sequence that brings about some sort of condition or disease, right? There is this sort of cultural temptation to still call it a mutation. If we want to get super technical, we could say a harmful mutation, we could say some sort of deleterious mutation. But the reality is that if you see a difference in genetic sequence that is behind a specific disease, no matter how rare or how prevalent it is in a population, we'll still call it a mutation, right? So I bring that up because it's, for most of us, before we start studying this thing a little more systematically, it is easier to associate mutation with something bad, with something harmful. What it's a little more rare is to sort of wrap our heads around the idea that something that is very rare in a population, we can still call a mutation. Now, from the point of view that it's so rare that it's just weird, it's a mutation, right? Just to settle the score, mutation comes from a word in Latin that means change. That's all it means. There's nothing in the word mutation that implies bad, right? But culturally, we've sort of grown used to the idea that mutations are harmful, that are undesirable. They don't have to be. They can also, there can be mutations that are simply rare in a population. Now, changes in DNA that are common but do not cause any clear dysfunction normally have a different name. So now we're talking changes in DNA or differences in DNA that are much more common than 100%. I'm thinking things like half this class will have one version of a genetic sequence, the other half will have a different version. We wouldn't call that a mutation because it's not having any harmful effect, but they're not rare either. So what would we call those? Do you guys know? It starts with a V. Variations? I'm not a good deep reader. Did you say variation? Yeah, variant. Close enough. Variants. So when you hear genetic variants, what we're talking about is a certain version of a genetic sequence that is not the same across individuals or across samples under study, right? So again, this class, there may be many genetic variants that are not rare. Half the class may have one genetic variant, the other half another, and so forth. And they're not clearly harmful either. And what do we call those genetic regions or we haven't really brought in this terminology strongly enough yet, but locus is any region in DNA, right? That normally we kind of equate to a gene. But strictly speaking, a locus is simply a specific place in DNA. So what do we call a locus that has several possible variants? Anyone heard of this term before? If not, you're learning it now then. Polymorphic. Poly meaning several, morphic comes from morphs or versions or forms of it. So it comes in several forms. And we apply polymorphic loci to refer to those genetic sequences that in a natural population will come in different variants. Okay? Questions? Concerns? No? All right. Let's move along. So, there's many ways to classify mutations because there's many different kinds of mutations. Two very common classifications are shown here on this slide. On the left, we are dividing mutations between somatic. Those are mutations, genetic changes in DNA sequence that happen in somatic cells and their descendants. Or germline. Those are mutations that happen in gametes, the germ cells and gametes produced by an organism. Super brief reminder, in any multicellular organism that undergoes some kind of sexual reproduction, the organism will have germ cells that give rise to gametes that those organisms use to reproduce sexually. All of the other cells in that organism would be somatic cells. In humans, the germ cells and the gametes would be in biological males the sperm, and in biological females the eggs, and all of the other cells in that organism are somatic. Okay? Then we classify mutations in these two categories that have to do with how much of that DNA sequence is affected by the mutation. So we talk about small or invisible mutations. These are point mutations or very small indels. Indels is an acronym for insertions or deletions. And we call them invisible because you would need molecular biology techniques to be able to appreciate them. There are other mutations that are actually visible and almost by the naked eye. So with a relatively simple microscope, you can see these mutations because these mutations are changes in DNA sequence that affect much larger sections of someone's DNA or something's DNA, right? So we're talking aneuploidies, which are changes in the number of chromosomes, or chromosomal aberrations that those mean, and we'll discuss them next week, those mean entire chunks of a chromosome that are missing, or entire chunks of a chromosome that are added to a different chromosome, entire chunks of a chromosome that are flipped. Yes?
Quick question. For somatic, for its descendants as well, can it also be passed down
No. So descendants, and that's why I'd like to emphasize here, its descendants. We're referring to the cell. So when we're saying descendants here, we're talking about the descendants of that somatic cell, not the individual, right? So The only genetic information that a human being can pass to its descendants would be through gametes. But if you have a mutation in a cell in your finger, that cannot be passed down to your descendants. I already explained. Good. So with that, I'd like to ask this question of the class. When it comes to somatic and germline mutations, which ones do you think are worse? Germline? Germline? Why? They get passed down, right? Yeah. Do they always get passed down?
No.
Whoa. Why?
Wouldn't the somatic ones be worse? Because wouldn't most germline mutations just not be viable?
Okay.
Or like really bad?
Okay. So a lot of things there to unpack. So when I asked, will all germline mutations be passed down? I heard like a collective no, right? Part of it is, yeah, not all germline mutations are going to be passed down. Why? Because randomness in the process of fertilization. So as human beings, we produce, millions of sperm, hundreds to thousands of eggs, right? But not all of them are used in making the next generation, right? So there's a component of a certain probability associated with passing down those germline mutations. With that said, if a mutation happens in the germline very early on, or if it is inherited from a previous generation, meaning that all of your germline cells contain that mutation, then your chances of passing that down are 100%, because all of your germ cells have that mutation. See, but that's sort of a more restricted case. More generally speaking, if you have an individual, person, animal, plant, and their germline suffers some sort of mutation, random, spontaneous error somewhere, the chances that that individual will pass it on are slim to very, very low. So in that sense, germline mutations are not sounding so bad anymore. Secondly, there is, sorry, I don't know your name. Sarah mentioned that many of these germline mutations lead to what we would normally call embryonic lethality. So there is obviously no procreation there is no next generation, but there is no suffering, there's no pain, no disease associated with any of that, right? Because there is simply an inviable embryo that could have been inviable for many other reasons other than mutations in the germline. So that kind of waters down a little bit the germline mutations. Yet we have this tendency to think they're worse because of that transmission component. What about the somatic mutations? What's wrong about them, or what can be bad about them? You have a mutation in a somatic cell. You're not going to pass it to your progeny. What could it do?
Well, for germline, there's a chance that can be passed down. Well, for somatic, it's there.
It's
So
there. It's there. It's there.
It's just there.
All right, so you have 3 million cells in your fingertip alone, maybe your whole finger, and one of them suffers a mutation. So what? Right?
It's going to replicate, or it might replicate.
It might replicate. It might replicate. And it might replicate with a mutation. So now you have 3 million cells all replicating. Now you replace the 3 millions with 3 new million, right? Because the cells that replicate often replace ones that are lost. There's still one that has a mutation out of the 3 million. So are somatic mutations a problem? I still see some heads nodding. So when are they a problem? Louder? Cancer. Cancer. Yes. Yes. Cancer is a genetic disease. And I don't know the statistics, but let's say it's educated guess 99.9999% of the cancers are spontaneous genetic mutations in somatic cells that make those 1 in a million cells not replicate once in every 3 years when they need to replace a cell that was lost from your fingertip. They start replicating when they don't have to. And their descendants that inherited the mutation will do the same thing. And their descendants will do the same thing. So now you have generation after generation after generation of cells that are constantly doubling, carrying the mutation that forces them to double when they shouldn't. And you get a tumor first and then a cancer. So this was just an intellectual exercise to get us thinking what mutations mean. From a species perspective, somatic mutations don't matter at all. But germline mutations do, because those might be the ones that lead to an adaptation, to a fixation in a population, to the wiping out of an entire species. I don't know. From an individual perspective, germline mutations often don't really mean that much when you think about it. But somatic mutations can mean the difference between living a healthy life and suffering cancer. Okay? All right. So, and then another classification we have for mutations is based on what they do, right? So there is some older but still in use nomenclature that classifies mutations as amorphs. So that means without a morph. And that's a slight misnomer in the sense that it doesn't mean that they don't have a form. It's just they leave the individual without that form, without that function. So amorph are mutations that take out the function that was normally carried out by that gene from the individual. There is hypomorphs. These are mutations that cause the function to be lost, but partially. So there is a loss of function, but it's not complete. Right? There is hypermorphs. These are gain of function mutations. We heard some of this nomenclature before. These are mutations that somehow enhance the function that was normally provided by that gene. Neomorphs, something completely new. So there's a new function. These are genes that are now doing something that they didn't do before. And then entimorphs, these are genes that are now negating the function that is provided by normal genes. And we know this as, let me see if I can, no, I can't, all right. We know this as dominant negatives. The idea here is that the concept of an entimorph or a dominant negative is particularly applicable to diploid organisms. It won't apply so much to prokaryotes that are haploid. So in diploid organisms, you always have two copies, two versions of each gene. If one of them is an entimorph, what that means is that not only is that mutant copy not working, it's also messing up the other. So that's why we call it a dominant negative. It's dominating over the other in a negative way. Okay? Questions, anyone? All right. So let's discuss the origins of mutations. Where do mutations come from? And this is slightly counterintuitive. If I ask where do mutations come from, many people may rightfully think some sort of toxin or mutagen in the air or X-rays and nuclear radiation and whatnot. All those do cause mutations. They do. But the majority of mutations that living things experience actually come from biological errors during DNA replication. Right? So DNA polymerases, when they're replicating DNA, they make mistakes. And many of them they correct. They have mechanisms to fix those errors. But some of them they miss. So what happens is, let's say that we have an original sequence here with a very specific AT pair somewhere in it. When that region is being replicated, the two strands will separate, and both of them are going to serve as templates for the new sequences. For one of these strands, the right base is paired up with the template, so a T is paired up with A. But for the other one, DNA polymerase makes a mistake and pairs up a G with that T. Once again, there are mechanisms or there are ways in which cells can detect these errors, go back and fix them, but sometimes they miss them. And when they miss them, the problem is that when that mutant molecule, DNA mutant molecule, will go and replicate, DNA polymerase has no way to know that that G there was a mistake. From there on out, it will think, DNA polymerases will think that that G was the right base. And they will perpetuate this mutation from that point forward. Right? So the next run of replication, when this mutant molecule separates the two strands and DNA polymerase sees that G there, it won't be able to say, oh no, this should have been a T. So I don't know why there's a G here. There should have been. No. It sees the G, adds the C, next thing you know, that thing fixed forever. Okay? So this is how point mutations or substitutions happen. Just small errors as DNA is replicating DNA, makes a mistake and pairs up the wrong nucleotides during DNA replication. We're going to come back to this idea of point mutations or substitutions. The name substitution is pretty self-explanatory. You've substituted a base that should have been an A there for a G. The point mutation, the idea behind that is that you have changed DNA sequence, but you haven't really changed the number of letters in that DNA. You've just made a point mutation. You've just changed one point in that molecule. A somewhat related and common source of error during DNA replication is a phenomenon known as strand slippage. So this is when DNA polymerase is reading through a DNA template strand and making a new DNA strand based on complementarity, as it should. And DNA polymerase runs into a stretch of repetitive sequence. So in this example, I'm going to zoom in briefly. In this example, DNA polymerase is reading through the sequence here, and it bumps into the stretch of GC, 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 G, right? So there is a strand of repetitive sequence. I used to do this. I'm not doing it anymore, but if I gave you a paragraph that all it says is the word house with no spacing, run one after the other, house, 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 and I asked you to read that, you would most likely find yourself reading, and then all of a sudden it's like you kind of stop and you go back, or you jump one, right? If I asked you how many times did you read house, you probably have a hard time. Same thing with DNA polymerase. DNA polymerase is reading through these things, and it is normal for DNA polymerase to sometimes become dislodged from the DNA template sequence. It kind of leaves the DNA template momentarily, but it can come back and continue. With these repetitive sequences, what happens is that sometimes that coming back is done in such a manner that DNA polymerase goes back in the DNA template and quote-unquote rereads sequence that it had already read, or jumps forward and skips a whole chunk of sequence that it hadn't read. Same as if I asked you to read house, 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 house. There may be one house that you skip or that you read twice. Okay? So here in this slide, what this slide shows is what would happen if there is no mistake. DNA polymerase detaches from the template momentarily, but then reattaches such that there is no shifting either way. And it continues and it generates a new DNA molecule that was the same as the parental one. But here we have the two examples of what could happen if it slips. So here we have, on the left, we have the case that when DNA polymerase reattaches, it does so jumping forward, and now it skips a couple of nucleotides in that original sequence. So the original sequence had 4 CG repeats, and now the replicated molecule has only 3. And again, we go back to this idea that this mutant molecule will be the parental strand for the next round of replication, and DNA polymerase won't know any better. Won't know that, oh, this one is missing a CG. It doesn't know. So it thinks that there should be a stretch of 3 CGs there, and from there on, it will continue replicating only 3. And here on the right, we have the opposite. So DNA polymerase detaches briefly, and then when it reattaches, it kind of goes back 1 position. So it reads twice the same CG. And now you go from 4 CGs to 5. And same idea, this change may perpetuate for many generations. So strand slippage is something that occurs when you have these sequence repeats that may confuse DNA polymerase a little bit, and it results in the contraction or expansion of the number of repeats. This is a cartoonish example in which the repeat is only CG. These repeats exist in DNA. They're a little more complex. So normally you have a repeat that is a little more complex than just 2 letters, right? But they do exist, and they do cause strand slippage, and they do lead to the expansion or contraction of sequences. So, real-life example. We take a slight detour to see applicability. There are these short sequence repeats in humans that throughout our evolution, throughout human evolution, were very prone to strand slippage, which led to polymorphism. Because throughout human evolution, these regions with SSRs either expanded or contracted. And they did so in a relatively random manner, but also led to the transmission of these variants. So here we have variant 1 with 5 repeats, variant 2 with 7, variant 3 with 9 repeats, and variant 4 with 12. Again, cartoonish, but not far from reality. And this cartoon here shows how some individuals will have one combination of variants, 1 and 3, others will be homozygous for 2, some other person may have 1 and 4, and so forth. These variants are floating around in the human population, and as humans mate and have progeny, they will shuffle these variants around and pass them on to the progeny. And we can use this because these variants are polymorphic enough that there is enough variance within human populations that now we can start drawing these very direct connections between individuals and their parents, or the other way around. So these SSRs, or short sequence repeats, are the things that are used in forensics or in paternity tests. We haven't done pedigrees yet, but a sneak peek. When you're drawing a family tree, circles represent the moms, squares represent the dads. So if you're doing a paternity test, what that means is there is a mom and a child, and then there is 3 guys who may be potential fathers of this child. So how do you know? So whenever you hear about this paternity test, this is what's going on. Someone will check the variance of mom and the variance of the child, and let's say that mom is 1-3 and the child is 1-2, right? So which of these is going to be the father? Well, mom could have passed down 1 or 3, and child has 1 and 2. So did mom pass 3? No. What did mom pass? 1. So mom passed 1. The child must have gotten 2 from dad. So now we go among these guys, blood test, see which one of this has V2. And that's how we answer the question. Who's the father? And in this case, who's the father? Number 3. Look out for a short writing assignment that I'm going to have posted over the weekend with a variation of this. But this is the essence. I don't know if you had come across this before or not, but this is the essence of paternity tests, of forensics. This idea that we humans are polymorphic for these short sequence repeats, They also go by the name of microsatellites. I don't know where that came from, but microsatellites. And we're polymorphic enough that the chances that we're going to find individuals that are identical for those combinations of SSRs within any random population of humans is very low. And that's why you can start drawing these, decent connections between parents and children, or like if I see blood in a crime scene, what individual or individuals may have come from. Yeah.
How would it be 3 not 1?
Because 1 could have passed 1, but we already established that the child inherited 1 from mom. Mom could pass 1 or 3, but he had to pass one of them. Because mom couldn't pass 3, because the child doesn't have 3, mom must have passed 1.
Sorry, I thought it was a 2.
Oh, that's a little one. Sorry, like if you zoom in, it's a little more clear. Yes?
Is this similar to the concept of codominance
In a way. So the question is this similar to the concept of codominance? In some way it is, in that you can quote unquote see both alleles when they're present in an individual. So we haven't gone there yet, but you guys probably heard before of dominant or recessive. And how we define those? Dominant alleles tend to mask the recessives. You won't see the recessives until they're with another recessive, right? Codominant means that if both alleles are present, you're going to see both. Cool. If I take a person, I cannot see what variants you have of a polymorphic SSR, right? But I can do a laboratory technique that we will study later in the semester to, in a way, find out if you have 5, 7, 9, or 12 repeats. So in that sense, I can see it. And if I do that on any of these heterozygote individuals that have one variant and then another, I can see both simultaneously in them. So in some sense, yes, this is related to the concept of codominance, except that none of these shows up phenotypically. You cannot, looking at a person, know what variants they will have. You have to draw blood, do PCR, and so forth. Yeah? If there are no further questions, let's move along. Where are we? 11. Ooh, okay. I'll speed this up. So, I already mentioned that biological errors are the source of most of mutations. Then I also brought up this idea of mutagens, right? Environmental toxins. It turns out that environmental toxins will indirectly act through biological errors. What I mean by that is most toxins, most mutagens, most things that we try and avoid, including things like UV rays and nuclear radiation, what they do is they actually mess up with some of the bases in DNA, either by modifying chemically or by themselves, sticking themselves chemically within the double helix. And when poor DNA polymerase is going through them, trying to replicate that particular position, they get confused. They see this thing in the middle and they don't know what's up, and then they add the wrong base. With UV radiation or other kinds of radiation, same thing. The radiation will cause chemical changes to the nitrogenous base that is associated to a nucleotide, and when DNA polymerase comes through, it will look at this base and, I don't know what goes in here, so then they'll stick any base, and chances 3 out of 4 is that they're going to make a mistake. Okay? So even these physical-chemical mutagens that come from the environment are going to indirectly act through biological errors. Yeah?
So is that how carcinogenic chemicals work?
That's right. So carcinogenic are really sort of a more impactful name for any mutagen. The real term is mutagen. They will cause mutations. Now, why do we say carcinogenic? Well, because many mutations lead to cancer. Does that make sense? There's no such thing as when you read the labels, this substance is known to be a carcinogen by the state of California. What that means is this substance is known to cause genetic mutations. And because we don't know where mutations might land, what kind of errors they might produce, might as well say carcinogen. Does that make sense? If you've ever been under the sun with no sunscreen, you've gotten some mutations. Now, the vast majority of them don't lead to skin cancer. So carcinogen is not like carcinogens are special kind of molecules that they know what to do to cause cancer. They just cause mutations, and then it turns out that many mutations will lead to cancer. Did that make sense? Yeah? All right. And then there's a third origin for mutation. This is a little trickier, and it's related to something that we call transposable elements. These are short DNA sequences that are somewhere between 100 and 10,000 base pairs. When I say 10,000, you're thinking, really? That's not short. But in the grand scheme of things, when you have a genome that is 3 billion base pairs, 10,000 is not that much, right? So these are short DNA sequences that can literally jump from one place to another in DNA. And transposable elements are sort of divided into two broad categories. Class 1, these are elements that transcribe themselves, and then through reverse transcription, that back arrow from RNA to DNA, will create a DNA molecule that then integrates, that's the term that we use, into the genome. So this piece of DNA will kind of paste itself in somewhere in the genome. The other class, class 2, there is an actual cutting of the original transposable element and a reintegration somewhere else. Strictly speaking, there is no change to DNA sequences in the sense that the DNA sequence in a transposable element is moving, but it's not changing itself. And same with class 1 and class 2. And there is no change to the DNA sequence elsewhere in the genome. So why are these transposable elements sources of mutations? They shift things over. That's one way to put it. So I said there is no change to the host genome. But if I move this, let's take the class 2. If I move this transposable element from here to here, there are two things that can happen. One is I may break something, and I have it on the next slide. I may, mutations can occur during random integration, so I may break something in the new integration site. Marker, please, thank you. I may be breaking something in the new integration site. I may be breaking something here. When these pieces of DNA integrate themselves into the genome, they may be interrupting something that was important, interrupting an important message. And now that message is not there anymore. Right? And then there are also some errors during excision. So when these things jump out of DNA for class 2, it's not a clean break. Often they will take with them a little bit of the host DNA sequence, and in doing so, they may be breaking something. Okay? So these transposable elements, show of hands, had anyone heard of transposable elements or transposons before? 2 hands. And I don't blame you. These are weird, right? Like so many other things, these are weird, and yet we're full of this. We're full of this, and for the most part, they don't move. They don't move. In evolutionary timescales, they don't move much. Every once in a while, they will decide to jump out, right? Or go through this process of transcription, reverse transcription, and reintegration. And when that happens, for the most part, nothing will happen because they may integrate themselves in an area of the genome that didn't really code for anything important. So we're good. But sometimes problems do arise. Okay? Questions or comments, anyone? We're good? Okay. So let's talk about what mutations do right? We've talked about how they happen, but now let's take a look at what they do. Let's talk about the effects of mutations. So small mutations, these substitutions, point substitutions, so the small indels that I was mentioning before, have diverse consequences. And those consequences depend on the type of change, right? So are we talking substitution? Are we talking small indel? And also where they happen in the genome as a whole, but also within a gene. There are certain mutations that happen in more sensitive areas of a gene, and the same mutations may happen in less sensitive areas of a gene. And we'll talk about the more sensitive areas now. We'll talk about some of the less sensitive areas next Tuesday. So let's talk about those mutations that happen in the protein coding region of a protein coding gene, right? On Tuesday, we talked about codons and how each of those codons meant an amino acid, and each amino acid in a protein is really important. Why? Because the sequence of amino acids will determine the folding of that protein in space and eventually its function. You change one or two nucleotides here and there in protein sequence, you change the sequence of amino acids, you may change the folding of the protein. Now, next thing you know, that protein lost its function, acquired a new function, has a function that dominates over the normal ones, any of the things that we discussed before, right? So protein coding sequence tends to be way more sensitive than other regions of the gene, or, of course, other regions of the genome where there are no genes, than mutations that happen in them. So, let's talk about point mutations. Turns out that when you change one letter in the protein coding sequence of a gene, when you change one letter, just the identity of that letter, the effect may be drastic or not, depending on where that mutation happened. So let's start with synonymous mutations. There are some mutations that just don't alter the identity of the amino acid coded by that particular affected codon. So the example that we have here, there is this codon here, the first codon in this stretch of sequence that they show us, TTA, gets transcribed into UUA. And if you look at the codes here on the right, UUA, I'm looking frantically for it. Where is it? Oh, here, leucine, right? So UUA calls for leucine. If this particular codon were to suffer a point mutation such that the A in TTA changes to G, now that transcribes to UUG. Guess what? UUG means the same, means leucine. So the genetic sequence changed. There was a mutation, but it's a synonymous mutation because the amino acid that is being coded for is the same. I'm checking the clock. I don't know where we got carried away. There's no way that I can get through the lecture. So what I'm going to do is I'm going to open the quiz for you to take home later. And you're going to have the whole day because I know that some of you have to go to class and whatnot, okay? So we can all breathe. We need to get through this carefully so that it settles, okay? Yes?
So a synonymous mutation changes the gene sequence, but it doesn't change the amino acid.
That's right. And I was having a conversation with one of you earlier. So I didn't catch your name before. Angelina. I was talking to Angelina during office hours earlier. When we use the term synonymous in languages, what we mean is different words that mean the same, right? Same here. So these are different codons that mean the same amino acid. All right? Going back to your idea of mutations that are detrimental versus beneficial versus neutral, what do you guys think a synonymous mutation will be? Will it be beneficial, detrimental, neutral? Neutral. It's pretty evident that a genetic mutation that doesn't change the sequence of amino acids will have no effect. It will be neutral. Okay? All right. Now, there are other point mutations that we call missense mutations. So now miss meaning different or like a deviation. So missense mutation. This change the amino acid coded by that codon, right? So let's go into this codon here. Originally, it was TGG transcribed into UGG, and that happens to code for tryptophan. A point mutation happened, a substitution. That T there was substituted for an A. So now AGG transcribes into AGG in the mRNA, and that translates to arginine. So we've gone from tryptophan to arginine. The amino acid changed. All right? So that is a missense mutation. Now we're missing the word. We're like saying something else. My question to you is, does that or do these missense mutations affect protein structure?
I guess it would depend on the ones that were swapped and what for.
It depends on the protein and what was swapped for what, right? That's absolutely correct. So whether a missense mutation with a change of amino acid will affect the structure of the protein and therefore its function will depend on where in the protein structure that change happened and what that change was. Why? There are areas of proteins that are not themselves involved so much in the folding of the protein. There are entire areas of protein that kind of float around in space. They can move freely, and you change an amino acid in them and it will force a change in the way that that particular stretch of the protein is oriented in space. It may get a little further away from some amino acids or a little closer to other amino acids, but it won't change the overall structure of the protein. Whereas there are other areas of proteins that are absolutely critical for the structure and the function of that protein. And the extreme example would be amino acids that participate in, let's say, the catalytic domain or catalytic sites of enzymes. I don't know how much you guys remember your biochemistry days, but in enzymes, these proteins that carry out chemical reactions, there are certain amino acids that interact chemically with the substrates and facilitate that chemical reaction between them, right? One second, I have a question before. So if you were to mess with any of those supercritical amino acids, then obviously that protein will lose its function, even though it may not necessarily lose its structure, but it will certainly lose its function. Yes?
Is it the structure of the new amino acid that makes the function of the protein change, or is it like a charge difference?
All right. So that's related to your second observation before, that not only it depends on where in a protein the change happened, but also what was swapped for what. Let's say that we have in a given protein a very critical aspartic acid. That's one of those amino acids that has a charge. It has a negative charge associated with it, right? And it may be, you saw it somewhere?
This one's arginine, but also
Arginine.
right?
It's yeah. So you have an aspartic acid somewhere in the protein, has a negative charge, and that aspartic acid is critical for interacting with a positively charged arginine somewhere else in the protein, such that these two regions of the protein come together. And that's very important for this protein, for its structure and therefore its function. Now let's say that this missense mutation changes that aspartic acid to a glutamic acid. Turns out that glutamic acid is negatively charged too, and it turns out that it can totally interact with arginine as well. So the protein itself may not care. So it's very, very difficult to predict. We have ways to make an educated guess, right? But it's very difficult to predict what the effect of a missense mutation will be. Because now we know enough to say this will change the identity of the amino acid coded by this codon. That we can predict, but we have no idea what the effect might be.
Yeah, that was actually what I was going to ask, because I was going to ask, like, if you change tyrosine to tryptophan, would that affect the structure as much as compared to, like, leucine to tryptophan?
We go back to the 2 points before. It depends on where you made that change and what those particular amino acids were doing. It's tempting sometimes to say, well, but like if I change, I don't know, a glycine for an alanine, they're both non-polar, small. Yeah, but if it turns out that that glycine had to fit in an atomic pocket within the structure of the protein, and now you stick a larger alanine there and it can't, then you might mess with the structure of that protein anyway, right? So it's very, very hard to predict what's going to happen. So, are missense mutations neutral, detrimental, or beneficial? Detrimental. It depends. Anyone for beneficial? They can, right? So again, this idea that an amino acid change, we've been talking about all these examples in which the protein might lose its structure. But what if that amino acid change makes that protein longer lived or more active or, I don't know, less amenable to targeting by a virus or something like that? Right? In any of those cases, that missense mutation could have a beneficial effect. So for synonymous mutations, we knew they're neutral. For missense mutations, they could be anything. All right, and finally, a very special kind of point mutation we call nonsense or asense mutation. That asense is a little older terminology, but you'll come across it in different places. Nonsense mutation, those are the ones that create a stop codon. So in our example here, the original codon was TGG. It mutates to TGA. That transcribes to UGA. And if we look at the codes here, UGA is one of the stop codons. So what that effectively does for this messenger RNA is when the ribosome is going through that mutant or nonsense mutation, it basically stops. And depending on where these nonsense mutation happen in that mRNA or in that genetic protein coding sequence of the gene, it may cut your protein short by only a handful of amino acids, or it may stop translation after 2 or 3 amino acids and basically have no protein. Did that make sense? So nonsense mutations, it's easy to think, oh, they have to be detrimental. They're basically taking out a chunk of the protein. Well, strictly speaking, it depends on where they happen. If you have a protein, some proteins have over 1,000 amino acids, and your nonsense mutation happened at position 985, well, maybe those last 15 didn't really do much for that protein, and that protein can still do its job without them. Same nonsense mutation, you place it at position amino acid 15, and essentially you have no protein. Okay? All right. Any questions or comments? Okay. So, all of that was about these point mutations or substitutions. Now, we'd like to start talking about these small indels and a very special effect they have that we call frame shifts. The idea here is as follows. Let's say that we have this wild type sequence, right? So it translates to this stretch of codons that will translate to leucine, phenylalanine, arginine, tryptophan, cysteine. That's cool. And then a small, in this case, in this example, there is a small deletion, right? So let me see what the original sequence was. So we had a TTT here. I can mark it with green. So we had a TTT, but due to replication errors, that T is skipped, taken away from the sequence, and from that point onward, DNA polymerase will keep replicating a sequence that is missing that T. what happens is the rest of the sequence shifts one position over. I emphasize the word shift because it's in frame shift. So there's a shift in the sequence. But there is also a shift in what we call the reading frame. Because remember that the ribosome is looking into the mRNA through that sort of small window of one codon at a time, right? Anytime that the ribosome has an empty A site, that A site is exposing 3 nucleotides. If you shifted the sequence by one position over, now that window will remain 3 nucleotides in length, but it's reading completely different sequence. So, I apologize for this, but let's try this exercise. We go from UUA, UUU, AGA to UUA, UUA, GAU. Even phonetically, you can sort of appreciate the change, right? So the whole sequence changed. It's not sounding anything like before. And that obviously results in a complete change of the amino acids being coded. Now you're not changing one letter, which may have an effect or not on the amino acid coded by that codon, depending on where that change took place. No. Now you're changing the entire reading frame due to these frame shift mutations, and the results are pretty disastrous because now you have a protein that has a completely different amino acid sequence. We go back to what we were just saying about nonsense mutations. It matters where they happen. You change the entire sequence of a protein early on, you've killed it. You change the entire sequence of a protein, the last 5 amino acids, maybe you can put up with it. Right? So where these frame shifts happen matters. So many frame shifts will lead to a wrong amino acid sequence that starts at the starting point of that mutation. This is something that I should have shown before. Anyway, so to illustrate this concept, let's say that we have an original message that is the dog can run. And I insert one letter. We talked about deletions before. Now let's do it with an insertion. I insert one letter where the E was. Between the T and the H, I insert an X. Right? So there's that new letter there. If I've been instructed to read words 3 letters at a time, now this sentence changed to right? Complete change in the meaning of that phrase. If I take out one letter, same thing. I'm changing the entire meaning of that phrase. Okay? Cool. Another common consequence of these frame shifts is the creation of a premature stop codon. So when you're shifting the reading frame in an mRNA molecule, those codons had the letters that they needed to have because they had been evolutionarily selected to have those combinations of letters because they coded for certain amino acids. Now, when you shift the reading frame, now it's almost like you're rolling the dice on these codons, right? Now they're going to have completely random combinations of 3 letters at a time. And it turns out that out of the 64 possible combinations, 3 of them are stop codons, right? So there is a slightly under 1 in 21 chances that with a frame shift mutation, you will spontaneously generate a stop codon. And 1 in 21 doesn't seem like much, but when you have a protein that has 1,000 amino acids, it will happen. Now, which one is worse? Doesn't really matter. Like, are you changing the entire sequence of amino acids in a protein or creating a premature stop codon? You've killed the function of the protein no matter what. But this is just to illustrate how not only can you change the identity of those amino acids, but sometimes you can even cause these premature stops. Now, everything that I just said applies to these small indels, these one nucleotide insertions or one nucleotide deletions. What happens if we have these insertions or deletions of 3 nucleotides or any multiples of 3?
It'll just insert or delete that particular amino acid and not affect the other.
Correct, right? So if we take out the exact 3 nucleotides that correspond to a codon, when the rest of the mRNA sequence shifts over, we've lost a whole codon, but we haven't really changed anything else. Now the ribosome reads from codon 1 to codon 3. But 3, 4, 5, 6 are going to be the same. Right? We can take the argument a step further. It turns out that I just said or emphasized what happens if the 3 nucleotides that are taken out correspond exactly to a codon. What if they don't? Well, it's funny. If they don't correspond exactly, you affect 2 codons, and then the rest picks up. And we'll see that in a second. And what I'm about to show for insertions applies to deletions as well. I'm sorry, what I said about deletions applies to insertions. So you can insert 3 nucleotides. If you insert them in something that we call in frame, meaning right in between 2 pre-existing codons, all you've done is add an amino acid. Maybe there's not much harm in that. You may be causing a lot of harm, but maybe no. Okay? But what if you insert 3 nucleotides in the middle of a pre-existing codon? I don't know about you, but my gut reaction is, oh, you're going to mess everything up. It turns out that, nope. What you do is you mess up that one codon that you inserted in the middle of, and the next, and then the rest fixes. It's a little weird, but it's true. So I'm going to go back to this cartoon example because the textbook didn't have examples of these in it. But let's say the original, we'll go back to the original, the dog can run. Right? So these are the 4 codons in this message, the dog can run. If we have an in-frame plus 3 insertion, meaning we've inserted 3 nucleotides right in between 2 pre-existing ones, let's say we insert the codon not, now the message changes. But it's not the end of the world. Now we're saying dog cannot run. Okay. It's still a phrase, right? We can still make sense of it. It changed, but we can still make sense of it. What about an in-frame deletion? So let's say that the whole of those 3 nucleotides in the first codon go away. So again, there's not much of a change. Dog can run. We can still make sense of that. Right? Here's where it gets funny. So what about those not in-frame insertions and deletions? Here, the not in-frame insertion happened right between DO and G. And we inserted the same word, right? The same word that we were inserting before, NOT, not. Well, we're inserting right in the middle of the word dog. What happens is dog breaks, but it kind of rescues it in the next codon. So you have the first 2 letters of the old codon, the first letter of the 3 that you're inserting, then the second and third letter of the 3 that you're inserting, and the third from the old. This thing affected 2 codons, but nothing else. The rest is fine. Is the message changed? Yes. Can we still make some sense? Well, the dog object can run. So there is, okay, I didn't get that, but there is something that can run. A not in-frame deletion, let's say that we take out EDO, leaves us with tug can run. Again, I didn't quite get that, but there's something that can run. Super cartoonish, but you guys see the point? So generally speaking, any 3 or multiple of 3 indels will have way less deleterious effect than a 1 or 2 or non-multiple of 3 indels, because the 1, 2, and non-multiples of 3 will cause frame shifts, whereas these guys will cause very limited damage. So now with these things, you're changing 2 amino acids in that protein. We go back to the same concept. If this protein had 1,000 amino acids in it, it may not be that bad. Oh, you guys saw to be continued and thought, all right, we can leave. Just give me 1 second. You're leaving about 10 minutes early. So all of that was substitutions, point mutations in coding sequence, protein coding sequence, right? But we also have, did you have this slide? Yeah, okay. But you also have mutations, same kind, point mutations or substitutions in regions outside of that protein coding sequence, before the AUG start codon or after the stop. What are those going to do? That's what we'll discuss on Tuesday. And yeah, that's that.